작은 창에 기댄 노을이 남기고 간 짙은 고독이 벌써 내 곁에 다가와 몹시 외로워져.
Okay, so now here in the mutation section, what you actually want to take a look at is what causes mutation in a specific way. How do we define mutations? At the DNA level, DNA small scale DNA sequence level, we are talking about. And what's the outcome of each different kind of mutation, and finally take a look at three actual examples, well-known examples of such a mutation. Then we end this chapter. So in general, mutation state definition is that the sequence change in the DNA. Okay. Why you don't really worry about any sequence changes in messenger RNA? There can also be a mutation because messenger RNA can affect the protein in the form of this codon relationship. But messenger RNA usually is made in very, very numerous, multiple copies. Out of a single area of the DNA, the number of copies that in a given messenger RNA corresponding to that area is like 100 and 1,000. So unless all those 1,000 copies of messenger RNA have exactly the same site area, same kind of DNA conversion, you don't really get to worry about, because there are, in terms of number-wise, it's just negligible. What's the probability of having any DNA, the sequence alteration in the RNA level? It's not likely. However, because DNA is your original single copy in many cases, then if you have this DNA sequence alteration, then now you have to take a look at more seriously. So any DNA sequence change. And then also mutation in actually more narrow aspect, that the DNA sequence change can occur in everywhere. However, in particular, what we are interested in is any DNA sequence change that has some kind of a consequence in the property of a gene is something that you pay any serious attention, is a usual way of looking at them. So three different types of mutation in that we are talking about a very small scale, although excuse me, the mutation can also occur in the grand, much larger scale in terms of the chromosome level that happens. So that huge segment of the chromosome can be deleted, broken, and then go somewhere else, stuff like that. However, first take a look at only very small scale of local DNA sequence level. There is one aspect, which is substitution, which is one particular nucleotide or segment of DNA is replaced by equal corresponding the segment in length or segment the same nucleotide, not exactly the identity, but if single nucleotide is replaced by another one nucleotide, in that case we call it as a substitution. Okay, and the other case is simply missing, a deletion. Okay, and also insertion. So you can distinguish these three differences. So in the messenger RNA, even though we are looking at the messenger RNA level because that's easier to understand in relation with the consequential amino acid change. However, we are not actually concerned about this actual messenger RNA level alteration, but this all has to happen on the DNA level and then affect the messenger RNA as a product. That's what we are talking about. So keep in mind that. So here one occasion, the example of a base substitution, one nucleotide A. Okay, originally there was a G guanine has been changed into A. Then now as a consequence, this codon GGC has been suddenly changed into AGC, which will signify different types of amino acid. Now it's a serine instead of a glycine. So this is an example of such a base substitution. How about base deletion? Now, one nucleotide, this one U has been deleted. Now, this odd thing happened in that once you have this missing U, then now all of a sudden why this original amino acid or whatever it is called a phenylalanine, is now changed into some other amino acids like leucine, and then from the point after, all those identity of the following amino acids have been altered into something. Originally it was glycine, but now it's an alanine, and originally it was alanine, now it becomes the other amino acids like histidine. What's going on over here? Here, what we can first figure out and then have understand the concept of such a reading frame is the thing. Especially in the case of this single nucleotide alteration of like either deletion or insertion, same thing. Either deletion or insertion of one or two nucleotide will cause this particular problem of, so we will see. So deletion or insertion of the one or two nucleotides will result in a frame shift of new ring frame. What is this? First, we have to understand what this reading frame means. And the reading frame is altered, shifted into the something is changed that we call as frame shift of the reading frame. So to do so, we have to first understand the concept of reading frame. This is one example of such a reading frame concept. Is although it is not something real genetic world's reading frame, but we can more easily understand in our own alphabetic sentence wise reading frame. So here, if you read, then it can make this sentence can make perfect sense. The red dog ate the cat. You know what it means, right? And then if you examine this, the structure, each vocabulary, each word, letter consists of 3 alphabet deliberately here. This sentence was made to illustrate the concept of the reading frame. So it's a THE, 3 letter, meaning one single particular word. RED, red, you know. So this is how it goes. But then kind of a frame shift occurs. So now you try to read. This was the original sentence that you try to read, and then probably it will be very difficult for any of us. Try it. The radoka teteka, right? It's very difficult. Why? Now all of a sudden the reading frame, which was originally making perfect sense, has been altered, destroyed. What happened? Look, examine carefully, and then you will see what happened. The guilty war is this guy. E has been deleted, omitted. So then, even with the deletion of this one, the middle E, that the maintaining the system of reading this sentences 3 by 3 is just keep going on. That's why this 3-letter reading frame somehow has been shifted to get this totally out of meaning sentence. So this is an example of the reading the frame shift. In the genetic world, the same thing can happen because 3 nucleotide will signify one particular amino acid, and then similar thing happens, like one single nucleotide is gone, and then cells do not know whether that single particular nucleotide is gone or not. So during the protein synthesis, it will still keep reading 3 by 3 packet. So that's an example of a frame shift. And you can try to expand this thing. So why that guy mentioned that 1 or 2 nucleotides of a deletion or insertion will generate this reading frame. Why not 3? You try to delete, instead of 1 single, but you try to delete 3 alphabet all together. Then now what? You try to read it. Then now the dog ate the cat. Even though now the new sentence become simply dog, not signifying whether the color of dog, but still it makes sense. The dog ate the cat. Same thing will happen in the case of this gene world. If you entirely delete 3 nucleotides, then the reading frame will be preserved. Okay? So that's why 1 or 2 may generate this whole chaos of the reading frame shift, but not the 3 nucleotide unit. And the example of the right is an example of an insertional reading frame shift. So here, the new the alphabet X has been inserted. So then now the original reading frame has been shifted 1 by 1. So then afterwards, total this reading frame does not make sense at all. Okay, and the same thing will happen in the case of a 2 letter insertion. But if you add the 3 letter, there's still a little bit of a change in the meaning, but still overall it maintains the communication. So that's the frame shift in summary. So that's the nature of frame shift mutation, and at the level of all these mutations we are talking about is a point mutation. Which means the mutation, the alteration of DNA sequence is in the scale of 1 nucleotide, is in particular this indicate this point mutation. But some cases like 1 or 2 or 3 nucleotide then still considered as a point mutations. So the consequence of this frame shift. Now there are 2 alternatives of consequence of this frame shift. As a consequence of this frame shift, either you have, and one possibility is having this so-called nonsense mutation. Nonsense mutation means you do not have any protein product. Nonsense. There's no sense at all. What at all? Okay, whatsoever. Why this happened is because as a consequence of this change, disruption of a reading frame, and very soon after the incidence of this frame shift, then spontaneously, by chance, the stop codon was generated. It's a lot of times it actually that's how it is. Even though you think there are only 3 different possibilities of a stop codon, UAC, there's 3 different UGA, UAA, UAG, 3 different kinds, but still it happens a lot. Once you have this the frame shift, then soon after, then one of those 3 stop codons are generated as consequences. And then this, from the start to the stop, is very short stretch as a consequence. So then we call it, then virtually no protein products will be made. So then now we call that as a nonsense mutation. In other cases, there is a missense mutation. With that, you carry on this damage and then you just keep all the way down, or some like some meaningful distance. And then in that case, we call it as a missense, because although it makes sense, but that sense made is totally wrong, totally different from the original sense. In that aspect, we call it as a missense. So wrong codons are all made, and then as a consequence, totally different and incorrect protein is made. Usually those proteins will not be able to survive in our this, the surveillance system of if there is any kind of malfunctions going on, we destroy. That's how we evolve. So anyway, the wrong different protein is the chance. Another actually often consequence is you survive. You just bear the brunt of this unfortunate instance. In this case, we call it a silent mutation. Even if you have this frame shift or whatever it has, or usually actually this silent mutation is almost impossible in the case of a frame shift mutation by the nature of this like codon in 3-letter nature of this codon. But substitution, a lot of times this substitution can survive in that you have no harm done to the integrity of the protein. Why? Because a lot of the codons are actually redundant. You have alternative codons signifying the same amino acid. The codon redundancy has one contribution. In another case, even if you have a change, actual change in the identity of amino acid, but still those changed amino acid can function normally in the place of that original amino acid if they were chemically similar in property. So same hydrophilic amino acid in the same position, or same kind of a dimension of amino acid, then overall you don't really have to worry about the overall consequence of this. A lot of times actually it does. So that is tolerant. This mutation somehow in this type of mutation is tolerant, so that maybe that is one of the actually room for the diversification of like making into the new version of your like functioning protein is this. So here the original and silent and nonsense and missense are kind of summarized here. So having said that, now we can take a look at 3 actual real examples of each different occasions of the mutation, each of which is actually very famous, especially in the Western world. One is sickle cell anemia, which is about the hemoglobin gene mutation, which I believe I have several occasions I have already mentioned in this classroom. It's called sickle cell anemia. It's actually caused by substitution, conversion of one nucleotide into something else. So in most of the cases, this substitution does not really cause any harm on the function or integrity of overall the metabolic activity of the cell. So in the mutation occurred in the beta chain, one of the 2 different kind of polypeptide chain of hemoglobin protein, the beta chain. At the early part of this the protein, the polypeptide, nucleotide adenine has been substituted to thymine, and then as a consequence, unfortunately, that single nucleotide conversion, a lot of times it actually could be tolerated by like signifying the same type of or same redundant codon, but in this case totally different amino acid So here E6V is one of the convenient way of describing the nature of this mutation. E here called one of the amino acid, the glutamic acid. Single letter convention of glutamic acid is E. Here the number 6 means that the position of the original amino acid. So it is relatively at the early head point of the polypeptide. The amino acid 6 position, which was originally occupied by glutamic acid, has now become another amino acid, which is valine. Now the problem over here is glutamic acid is an extremely hydrophilic amino acid, whereas valine is a hydrophobic amino acid. You got a problem actually here then. This is overall shape of hemoglobin protein. Two same colors is over there probably beta chain, and another same color is an alpha chain. Each of which of all these 4 subunits can pick up single oxygen molecule. And then as a consequence, this whole the correct folding pattern, which is important for its proper function, is now totally become differently shaped, differently folded protein, which even pile up inside of the red blood cell, like this. So if this is a normal shape of like a ball-like, we call it as a globular shape of hemoglobin. Now, after there's such change, now this causes a really chain of a clumping of all these different hemoglobin protein, and they precipitate down in the bottom of the pile of stockpile of red blood cell. Actually, there is a reason why this hydrophobic and hydrophilic nature is really something to do with this conversion, because usually those hydrophobic amino acid, I'm not sure whether if I really should go into this kind of things, but just for if you are kind of curious, because actually this type of protein clumping due to the mutation is actually everywhere. Not only is in the case of the hemoglobin mutation in the sickle cell anemia, but in many other occasions, including diabetes and even the Alzheimer's and even Parkinson's disease, and all actually can now ultimately, if you look up the reason, then it points to this protein clumping due to the structural change. Same thing. And this is one example. And so for that aspect, I will just try to mention the reason here is usually the hydrophobic water-fearing part of the entire protein will try to hide inside of your whole entire protein body, and that's one of the major driving force of spontaneous protein folding. So like once you have this newly made fresh polypeptide chain is produced, and they spontaneously fold into some shape. Due to this, some of the hydrophilic, the water-loving amino acid will cover the surface of this protein, and those hydrophobic protein will go inside to avoid any water contact. That's the most energetically stable structure, and that's why a specific protein is made into certain specific shape, which is relevant and critical for its respective function. But now you have a different hydrophobic protein exposed. Normally you have that position, the hydrophilic protein must be there, but now due to the mutation, now this guy is. Then what are you going to do as a molecule? As a molecule, you extremely hate, abhor any water contact. The only possible way of avoiding such water contact is just like the case of this spontaneous formation of phospholipid bilayer. Same thing. These guys, this hydrophobic area will try to stick together so that they can cover up themselves from any contact from by the outside water. That's how this hydrophobic residues connections generate this kind of clumping. And same occasion, same examples can repeatedly be found in other such similar occasions, and then affect very seriously on in terms of the disease symptoms in many different occasions. So here the ultimate occasion is the sickle-shaped red blood cells, because the mature red blood cell nothing but contains all whole stocks of hemoglobin protein. But if your hemoglobin protein structure is something malfunctioned, this the clumped one, then actually you are as a container, your shape will also be deformed. And not to mention that inside of this shape, deformed the cell, the actual functioning unit of this hemoglobin protein cannot provide any actual function of oxygen carrying. So you are in serious problem. So that's why it is the overall disease name is called the sickle cell anemia. Anemia means the lack of the oxygen. Actually, anemia means a lack of blood, but it's not actually the lack of blood. Your red blood cells are just the number wise, it's perfectly fine, but only simply they become obsolete is the problem. And this was the one that I just mentioned, that clumping is made. This instead of a valent substitution, now what you have is interlocking of each those hydrophobic residues. So as a consequence, you have this type of sickle-shaped red blood cells of out of function. So not only this oxygen carrying ability, but so many other functions are also affected by the sickle cell anemia. You can easily guess why, because this is one of the most critical function, the oxygen carrying. So if you cannot provide the proper function of this, then almost everybody will be under the influence of this defect. So that's what it is. So such example, now you have this particular trait was the result of this one particular gene, the beta globin gene mutation, a single mutation. But as a consequence, what you have is all those many different other areas of the symptoms. This type of one gene, as a result of mutation or some trait, one trait affecting many different symptoms. Okay, many different traits, we should say, different characters in the form of specific trait that we call as a pleiotropy, multiple occasions of pleiotropy, like this. So that's pleiotropy. However, the other way around, the opposite, you have only single characters as one specific trait, like human skin color, your eye color, or even your intelligence, height, body weight, many. So there are so many different occasions that each single trait is the result of actually multiple. It's not a single but multiple genes interactions. We call that as a polygenic inheritance. So this is a totally kind of a contrasting. I wouldn't say it is an opposite, but it's a kind of quite contrasting examples of one having multiple influence and the other one has many having only one single particular characteristics under its influence is this. And now this sickle cell anemia is a recessive mutation. What is the recessive mutation? Once again, we'll just bring back our the memory for this discussion of actual why what is recessive mutation? Once again, is if you were asked that what is a recessive mutation, then it's easy. A recessive mutation by definition is that as a trait, when you are present in heterozygous status, then your trait is hidden. The other guy, my alternative guy, will take over, cover up my whatever trait. Then now I am a recessive in terms of a recessive trait. So this mutation will result in a recessive trait. In other words, if you have your two homologous chromosome, okay, and one copy of your beta globin gene is actually affected by this particular mutation, the sickle cell anemia mutation, but still the other part, the other copy partner on your other homologous chromosome is normal, perfectly does not have any such mutation, then outside, phenotypically speaking, you are almost perfectly normal, almost. Okay, so this is called. So then it is the other guy is a dominant, and this guy, the sickle cell mutation, is recessive, right? This another interesting thing about this heterozygous status in this sickle cell anemia is, even though as a heterozygous having one copy of your gene at least is carrying this really serious mutation, but in some geographical region, now actually carrying this mutation as a heterozygous is a kind of a selective advantage. So here, the big alphabet letter, two big letter alphabet A signify this two dominant, which means non-mutant, normal beta globin allele. Okay, here one little a signifies this mutant, sickle cell mutant, and the other one is so-called normal. I should be careful about referring to normal, but normal, let's just, for the sake of convenience, let's put for a moment it's a normal. So, in this case, it's a heterozygous, and the symptom is as an individual carrying this heterozygosity of the sickle cell disease mutation, in maintaining the everyday routine of life, it's just perfectly okay, unless you have somehow decided to live for the rest of your life in the up in the high altitude, like in Himalaya, where the oxygen supply is extremely scarce. Other than that, you will be okay in most of your daily everyday life. But one extra benefit in where, depending on where you are, is this malaria infection. This guy shows much stronger resistance. Yeah, I'm just kind of curious that whether if this heterozygosity can also be like resistant to any kind of other viral disease. Like, although malaria is not a viral infection, but into the coronavirus infections, stuff like that, it would be really interesting to see. But anyway, and this homozygous for this recessive, the mutant allele, you have a really serious problem. It's a fatal, or in most of the cases, it's a fatal. You're not going to be able to live that long with this type of serious defect. If that is the case, the reason why we bring up this is this. If this was something that, otherwise, if this one does not carry such kind of extra surprising benefit of advantage of being resistant more to malaria. By the way, I also know that the actual mechanism why this one has a resistance to malaria, and I used to go over this actual mechanism like several years ago in this same classroom, but not anymore. So forget about that. So then, in the population where in the region where the malaria is such a serious problem, then you will get to have unusually high proportion of this heterozygous individual. Otherwise, the fact that during the gene segregation, you just keep producing your offspring, sons and daughters, and then some of them will emerge as in this form of this carrying two recessive alleles. Then the minute you have, then this gene will disappear in the population pool, as you can easily imagine, because this person is not going to be able to live long and then to be able to produce any viable offspring. So then more and more the frequency of this the mutant allele will go down, right? However, in this particular area where this malaria has been a serious problem, and then you relatively have such a high proportion of the individuals still carrying this heterozygosity. Okay? So usually this black, especially this African black population, tend to carry this higher percentage of this mutant allele is the reason why. And this is a historical distribution of this malaria isn't exactly matching, isn't it? So once again, revisit this point of emphasis about why is the sickle cell allele recessive? Why? If you were asked why, then you should be able to provide not. So now just forget about well, that's because it's when they were heterozygous status, and then this sickle cell allele is not going to be shown. That's why it is recessive. No, that's what the recessive is. The reason why, in terms of this molecular mechanism, is because it has lost its function, right? This sickle cell allele cannot provide any function. This is as a result of this mutation, the original function of carrying the oxygen by the hemoglobin is lost. So this can be characterized as loss of a function. Then anytime you see this loss of function, then it's automatically it will be recessive mutation, because when this loss of function mutation is present together with the allele carrying the normal original function, then that still the original function will be able to be provided by that guy. So nothing to show over here. It's just everything is normal, at least on the surface. That's why it is recessive. And once again, as we went over earlier this section, this chapter, then why some mutant still, unlike this sickle cell anemia, some other type of mutations like Huntington's disease, it is actually dominant. So in other words, even if either one of your two copies of genes has become this mutant, then you are in big trouble, because that one will take over and then show it's the different this disease related symptom. Why? Because in this case, this new mutation resulted in a kind of a new function. So once again, a new function, or kind of interference of existing function. Can also play as a dominant allele, because, for example, like in the case of a sickle cell mutation, imagine if that mutation played in this way instead of simply providing a loss of function. But what if this converted altered beta hemoglobin structure is somehow inserted into existing this fine structure of a 4 polypeptide subunit, and then once get in and then totally disrupt even the normal hemoglobin protein, then you are in a big trouble. Actually, the similar example is where the mad cow disease, okay, the protein prion is doing exactly very similarly such kind of the function in such a way. Okay, So the second example we are going to take a look at is a cystic fibrosis, which is actually for those Asians like us, a disease that we don't really have to worry because the incidence, the frequency of this mutation in this ethnic race population is extremely rare. But on the other hand, this white Caucasian population of a certain origin, this the frequency is extremely high, so that they have practical. It's a kind of practical concern, and it is this reason why in almost every the biology textbook, which are all which all come from those like in the Western world, they make sure they always mention this cystic fibrosis as one of the major, the case of important in mutation, and that's the reason why. And the cystic fibrosis, look, how many different. So this is one of the such a pleiotropic the mutation. So many different areas is being affected by cystic fibrosis, and the protein responsible for this disease is called. Its name is a CFTR. Abbreviating this is rather a long name, right? Cystic fibrosis transmembrane conductance regulator, whatever. This is a membrane protein on your cell membrane, especially abundant in this area, respiratory tracts, and description of this, the characteristic description of this mutation can be described like this: delta. This triangle means a delta, which means a deletion. Delta F508 means this F is one of the such amino acids, a phenylalanine. Number 508 means an amino acid, the position 508. So this CFTR protein must be really big protein, right? It is. And at the position of a 508th position of it, then lies the particular amino acid named phenylalanine, and that's been deleted entire 3 nucleotide has been deleted. So as a consequence, this one single amino acid has been gone. So to our common sense, in this occasion, we can anticipate that nothing serious would come, because you still get to preserve the reading frame, even though you just lost one amino acid. But so what? It's just one single amino acid out of how many really big, in really big protein. How many amino acids are there? And then it's just only one amino acid. You won't even notice. But this time you are in a really bad spell in that this single amino acid disappeared, makes such a night and day difference in that total protein folding happening in your inside of your endoplasmic reticulum. Remember, in the pre-Milton era, and then totally messed up. So you don't have any correctly folded protein produced, especially if you were homozygous for this, which meaning no functional copy of genes is present. So all the protein made will be just incorrectly folded protein, and those incorrectly folded proteins are all destroyed inside of your endoplasmic reticulum. So on your cell membrane, no such CFTR proteins are present. You can find in such a patient. Is it really important protein? Yes, it is. This is CFTR function as a kind of a transporting protein. What does it transport? Ion, like a sodium or chlorine kind of ion. Like sodium or chlorine type of ions are being transported through this particular protein. And this is overall cartoon describing the shape of this CFTR protein on the cell membrane. And the importance of this CFTR protein is by doing so, this pumping the ion, the consequence is it keeps the liquid, the flow of the liquid outside of this tract going on. So with this water flow and this ion traffic, then you can somehow shed off with your external surface of your this respiratory tract, where all these cilia. Remember, this is one of the sources, cytoskeleton protein, cilia and the flagella, and the cilia is really so many short hair-like proteins on the surface of your many different tissues, right? Even I mentioned that if you're a smoker, the cilia on your respiratory tract is all destroyed, so you will have a serious damage, and you will be very vulnerable to any kind of a general infection because of that. And so is this case. So, due to this lack of water flowing, and then as a resultant of this ciliary movement, just like all these the grasses on your field to the wind. Because of that, all those thick mucus much thicker mucus will build up, and it's like a mud. And within this mud, then you can easily imagine that a lot of those microorganisms can be trapped. So that's why this person will become so vulnerable, sensitive to this infection. So because of this, the ion shooting out, and then the resultant of this water flow maintenance, and you have this thick mucus built up, and as a consequence you have such really will become a person which is very sensitive to any kind of a pathogenic infections. So basically all those different kind of pathogenic microorganisms will have established some kind of a film over here. Yeah, that's not good. And luckily it is a recessive mutation, and you know why. Okay, you can easily understand why it is, because as long as you have one functioning copy of this gene, then that copy will be able to generate the normally folded protein, and then they can end up on the cell membrane and then provide the function. That's why. And the frequency is extremely high in that in European heritable populations, like in 1 in 3,000. Wow, that's such a 1 in 3,000 of this homozygous the incidence of this cystic fibrosis, which is really high. And finally, let's take a look at another type of mutation, which is now in this case is a dominant mutation. It is also caused by a base substitution mutation. The two interesting characteristics with this particular mutation, the name is lactase persistence. One is, even with this the single base substitution mutation, it is a dominant mutation. The other one is, this mutation, the base substitution, is not something that takes place in the coding region. It is non-coding regulatory region, far from the actual transcription start site. And then you will still have this very much noticeable consequence. Here, the enzyme lactase. This enzyme lactase is responsible for allowing us to be able to digest any dairy product and a milk-containing product like some milk and ice cream. So this is very important for me personally. The person like crazy about ice cream. So I am, which is very important for me. I'm serious. And this lactase is the one that break down. There is a specific type of sugar, lactose, present in the milk of these mammals, like cow milk and human milk, all those lactose is present. Now, those infants, whether you are calf or whether you're a baby, human baby, now as an infant, this lactose sugar must be then the major source of your nutrition, because you don't have enough viable digestion system for other food, like heavy food, so that out of which you can digest all those grains or any other meat, so that you can utilize them. No. Then what are you going to do? So you have to be heavily dependent upon this enzyme lactase when you are early, like early baby infants. Yes, that's the case. However, our modulation system is an example, another example of such developmental specific expression of this, the lactase is. So this lactase expression in transcription goes down significantly over ages, so that when you reach something like a middle age, you lack of. A lot of people simply do not have any lactase expressed. That means then now you can no longer be able to digest this any dairy product. So then those people will have a problem whenever you just consume the dairy product, and you have this build up in the uncomfortable gas. Actually, that is exactly the case of my wife. So both we together, when we were young, not beautiful, but when we were young, we were able to spend the whole night, stay up all night, and then spend the whole bucket of ice cream together and stuff like that. But no longer we can enjoy it because she has a specific trouble. She cannot, but I do still. I can still have no problem over this digesting any dairy product, even with this old age. Okay, why? Because some people actually carry this mutation. So in this occasion, due to this mutation, now you obtain the ability of this sustained expression of lactase. So it's just the choice between whether you do not have any transcription of the gene, or you still have, at the late developmental stage of as a person, you still have this lactase expression. Then now you can have this function. So now you can understand why this occasion of the mutation is a dominant mutation, right? Because it is now the obtaining a function versus the disappearance of the function. And another very interesting aspect of this is, as I mentioned, this mutation does not take place on the coding area of the gene, the enzyme gene. It's one single mutation, point mutation in enhancer region, lactate gene. But even this is shocking in that the distance between the transcription start site and then this particular enhancer region is, wow, look, can you see? 14,000 nucleotide. Oh my gosh. So how can you actually imagine, there's such a long distance, any area this can have any kind of influence on the gene expression? Because if this was the area, by the way, this point mutation is a this C to T conversion. Originally, there was one nucleotide cytosine was there, but those who are able to now express still the lactase at the old ages, this particular point has been converted into T here. But anyway, whether it's a T or C, that means simply this area still can be in the mutant individual. This area can still be occupied by some specific transcription factor, and from such a long distance, which is a 14,000 nucleotide, can now somehow influence this. And how? Probably we have seen the mechanism by when we were looking at the mechanism of this tissue-specific expression in the case of this albumin and the the crystalline protein earlier today, right? So with that, I think the dominant mutation, and why and this mutation is also highly frequent in the Caucasian population, because culturally speaking, those people were actually heavily dependent on their everyday living on in hunting and also this the whatever the livestock they used to live much earlier than historically speaking. They just maintained this agriculture with this many various livestock, unlike heavily those Asian populations concentrating their activity in the sheer agriculture of this grains. So like Asians, in particular East Asians, and then American, the Native American in the United States, and I mean the America. I shouldn't point specifically only United States. That's my it's wrong. So this population has a specifically very low instance of this mutation, and so like it's kind of a personal surprise that still I do have no problem in this ability. So this is something that shows how like the darker this color, showing that the higher the frequency of this mutation is in small scale. Okay, so with that, we think I think we can finish, although with a lot of those the asterisk and then regret, we stop here our these lectures, and like I said, exam will cover only up to this point. So good luck.